So yesterday I showed you how to calculate the surface, the volume, and the surface volume ratio. We were talking about cubes, but realistically we're going to apply that to cells. That's going to be the topic that we're going to relate it to. And I mentioned the word metabolism yesterday, and so today we have to talk about that part. Now most cells range in size from a micrometer to a millimeter. And a millimeter is really, really big for a cell. Most cells are not that big. If you think about the cells that you observed in the lab, for example, on high power, most of you could see in the neighborhood of like 100 cells in your field of view. And if you're thinking about how that's not even half a millimeter and you could see 100 cells, you get an idea of how small each of those cells really is. So most of them are very small. So usually we measure the size of a cell in micrometers because most cells are so small that a millimeter is too big to measure them. The size of the cell is limited by metabolism, and then these points underneath it explain what that is talking about. Like I mentioned yesterday, the word metabolism is talking about chemical reactions and processes that go on in the cell to keep the cell alive. And so an excellent example of metabolism would be cellular respiration. Now I'm mentioning that process because we already know about it from learning about what a mitochondria does, but that's an example of a process that helps to keep a cell alive. And so in order for metabolism, a chemical reaction like cellular respiration, to be maintained, nutrients must pass through the cell membrane. The cell membrane is the surface of the cell. So when we're measuring the surface area of a cell, what we're really measuring is how much cell membrane there is. Now, examples of nutrients that have to get into the cell, for example, would be oxygen or glucose if we're talking about cellular respiration. Cellular respiration can't happen unless oxygen and glucose are getting into a cell. And it needs to keep on entering the cell continuously. As soon as we run out, cellular respiration can't happen anymore. So the idea is that if the volume of the cell is greater, then so is the metabolism needed. By that I mean the amount of reaction. So the bigger the cell, the more cell respiration that needs to occur to keep it alive. Now, as cells grow bigger, what we'll see is that volume increases faster. What we mean by that is that if I were to make a cell bigger and re-measure its volume and its surface area, they would both get bigger, but the volume would get bigger by a larger amount than the surface area. So when we say faster, what we really mean is by a larger amount. And you guys will get to observe that in the lab that you're doing, because you'll have things that are different sizes, and you'll notice that as we increase the size, the surface area and the volume don't change by the same amount. That means there's a problem for larger cells. When a cell gets so large that the surface area, so the membrane, cannot supply nutrients fast enough for the demands of the volume of the cell. The cell could not grow any larger in that case. Now there are some things that we could do to increase diffusion. And so sometimes even large cells can exist if other things are happening to make diffusion happen faster. For example, heat usually works. If we heat up particles, they naturally move faster, and so that could increase the rate of diffusion. if we make smaller particles, that could help increase the rate of diffusion. Smaller particles can move straight through the cell membrane, whereas larger ones usually need a protein. We could also have a larger concentration gradient. All of those things could help a large cell have more transport across the surface area that it has. But even with those three things, we would still reach a point where I cannot get things in fast enough to supply the demands of the cell. So very, very large cells, fat cells, would be an example of a very large cell, are generally just for storage. And they would have very, very slow metabolism. That means that not a lot of processes would be happening inside that kind of a cell, not like, say, a muscle cell that's constantly contracting and relaxing to help you move. The way that we'll determine how big a cell can get is really the surface area to volume relationship. So we saw yesterday how to calculate it, and so that everybody is clear, bigger is better. When we calculate the surface to volume ratio, it's always some number to 1. The bigger the number, the bigger the surface to volume ratio. So it's desirable, for example, to have a ratio of 5 to 1 compared to a ratio of 0.2 to 1. So the bigger that first number is, the better the ratio is. This is the reason that cells can't be very big, the reason that a millimeter is about the maximum. So what I'd like to show you are just a couple of examples of actual structures or cells in living organisms that have a maximized surface to volume ratio, like they've really gone to a lot of trouble to have tons and tons of surface compared to their volume. So I'm just going to show a few pictures, and then I'll come back to this screen with words in case anybody still needs it. These are 3 pictures that are examples of maximization. By that, I mean they have very large SV ratios. Now, does anybody know what any of those pictures are of? So I'll tell you, the first 2 are inside of you. Yeah, George, you know one? The last one is definitely the root of a plant. That's what it's a picture of. Anyone know what either of the first 2 are? There's even a little faint word here. I don't know if alveoli would clear things up for people. Capillaries is actually a good example too, but that's not specifically what an alveoli is. Someone else was saying they knew what an alveoli is. Yeah. It's in your lungs. So this is a picture of alveoli, which are in your lungs. Anybody know what that middle one is? Yeah? That is your intestines. So this is a picture of the inside of your small intestine. Now, I would ask you on a quest, what's an example of something that has a maximized surface to volume ratio? And these would all make excellent examples. So that you know, alveoli are the individual little sacs. They look like little bubbles that are at the end of the tubes through which air enters your lungs. So, this is called a bronchiole. It's a subdivision of a bronchi, which is attached to your trachea. The trachea is the thing that goes down your neck. If you take Bio 20, there's a whole chapter just about this. But so that you know, at the end of every tube through which air enters, instead of it just being, let's say, a flat surface like that, it has these tiny little bubbles on it that make it look bumpy or rounded or wavy. This bumpy or wavy look is usually what you're looking for if you're trying to find something that has a maximized surface to volume ratio. What it means for your lungs is that instead of oxygen and CO2, so oxygen is coming in, CO2 is coming out, instead of them only being able to be transported across one flat piece, now we've made all of these little bumps. If I were to stretch that out, it's actually a huge amount of tissue. In your small intestine, you'll notice that right away it has kind of a wavy structure. Then it has these little projections, and on each of those projections there are other little projections. We often refer to the inside of the small intestine as a brush border. It kind of looks like a hairbrush with a bunch of little bristles on it. If we were to stretch this out, it is incredibly long. And so your small intestines fit in your abdominal cavity. They don't seem like they're very big. But if we were to take all of those folds of tissues and stretch them out, you have like kilometers of small intestine built up inside your body. If I was to take every little wavy bit and stretch it out, your small intestine is actually 600 times more surface with all of these waves than if it was just a straight solid tube. In your small intestine, things that are going sort of from it out into your blood would be all the nutrients that you have digested and that your body is going to absorb. So earlier Isaac mentioned the word capillaries. Around your alveoli and around all of the parts of your small intestine are little tiny blood vessels called capillaries, and they surround all of these little folds, making it easier for your body to absorb what's there. On the root of a plant, there are little hairs, and they have the same function as the alveoli or the small intestine. If I were to stretch out all the tissue that made up the root of a plant, it would actually be very large. So the hairs are little projections, just like you have in your small intestine. These are all examples of lots and lots of surface for a relatively small amount of volume. So I'm